All right, so this one here, which is not one that we talked about yesterday, but was one of the ones that I set for homework. You've had a look at it?
I haven't done it though. I couldn't do it.
Same.
Okay. Well, let's work our way through that, because obviously building that skill and building some confidence around that is going to be important. This question is well, you tell me, what's the question really focused on here? So it's to do with pathogens, but what's the question? What are they really asking you about here? So, well, let's work our way through. We'll work our way through, because I'm not getting a lot of answers there. We'll work our way through and we'll see how we go. I might just flick this up if I can to my other screen, just so I can actually see you guys at the same time, because I couldn't tell if you had left the room or anything then, because I couldn't see you. All right, so Sharon wanted to investigate the effective increase in the concentration of medication against the growth of an unknown pathogen. For her experiment, she prepared 5 different concentrations of the medication. Yep, you can read those, which is known to kill pathogens by interfering with the production of their cell wall. 5 agar plates containing each concentration of the medication and the pathogen sample were set up and left for a week. For her experiment, Sharon found that the medication had influenced the growth of the pathogen. Plates that had a higher concentration of the medication had lower rates of pathogenic growth. So based on the information provided, what 2 kinds of pathogens could Sharon be using in her experiment? So that one's specifically about the nature of pathogens. That's what the question's asking you. It's related to this chapter. The other stuff isn't really asking you about pathogens. We'll come to that in a sec. But for that one, work your way through what you know. So what are the 2 broad categories of pathogens?
Non-cellular and cellular.
Non-cellular and cellular. So what category are we talking about here? Don't worry about naming them. What category?
Cellular.
Yeah, because it interferes with their production of a cell wall. Well, it has to be cellular, right? So I can exclude what? What are the 2 that are non-cellular? It's only 2 that are non-cellular.
Viruses.
Yep, prions and viruses. Great. So we know it's not those. So realistically, if you named any of the other 3 that we talked about yesterday, you need to name 2 of them. But if you can name 2 of the 3, you're going to tick that box. So what's one?
Fungus.
Yep, absolutely.
Worms.
That would be one. Sorry?
Worms.
Yeah, worms. Or what's probably the most common one? That's the one that I think of when I read this, but then I did work in the microbiology lab. Yeah, bacteria. Yeah, that'd be the most. What I'm thinking when I read this is it's an antibiotic and they put it into the agar. That's because that's something that we actually do. But that's what I think when I read it. So identify a possible control. What could be a control for her experiment? So now we're going into experimental design. So if you were designing this experiment, what would you use as a control? So think about what you did with your photosynthesis experiment. You had controls in that. What would your control be here?
Would you have one with 0?
Yeah. Just a plate with nothing in it. Because, well, that's the next question, so I better not say because. Why would having a control be important?
To make sure the experiment is valid.
Yeah, that's absolutely right. So be specific though with this though. So you've got what you said for your controllers, you'd have one with 0% of the medication. That's fine. What would you expect the result to be?
Nothing.
Nothing.
What do you mean nothing?
No, it would grow.
Yeah, what if it didn't?
Then the experiment didn't work.
Then the experiment's invalid, right? So you need to check that the pathogen actually does grow in the agar. What if she selected an agar that actually is incompatible with growing the pathogen? Again, having worked with bacteria for 20 years, you don't need to know this, but I can tell you if you put the wrong thing on the wrong plate, it just won't grow. So she might have picked the wrong plate. If you didn't do a control, how do you know that it's the medication that affected it and not actually what was in the plate? So yeah, that's really important as well. While observing her results, she discovered another pathogen growing on the agar. Suggest where it may have come from. That's an interesting question.
It could have already been in the agar.
Yeah, it could have contaminated the agar. That's fair. And really any reasonable, this is the sort of question where it's going to be any reasonable response would go, yep, that's fine. So that could be it. She may have contaminated it with something that she had on her. So it may actually have come from Sharon herself as well.
Could you just say contamination?
Yeah, contamination from either the plate, contamination from her skin, contamination from the air, like it could have come from anywhere. Poor technique would be the take home that I would use for that though. Right, so that's, but that's good for that one. It's not sharing that screen. I've brought up the other questions, but it's not sharing it. So bear with me a second. But again, that one, it's designed to take information that you're building on throughout the course of the year, right? So it's not purely about pathogens. Part of it was, but it's also about experimental design and work that you've done leading up to that point. And this question is going to be similar. We touched on this one a little bit yesterday. So well, you guys can tell me for part A. Well, let's go through it. Salmonella is a bacterial pathogen, causes food poisoning in Australia, usually occurs after eating contaminated food, but sometimes after contact with an infected person. Symptoms include diarrhea, vomiting and fever, usually lasting for 2 to 7 days. So part A, recent evidence suggests that normal flora in the gastrointestinal tract may provide protection. Describe how normal flora can protect against salmonella. So it's only worth 1 mark. You don't need to say a lot, but what would you say in response to that question? So did you have a look at this one page but haven't answered it?
Yeah.
Okay, so I'm going to lean on you to answer this one a little bit.
I don't know. I didn't look at the answer.
No, you don't need to look at the answer. We talked about it yesterday. So when you've got normal flora, so remember we talked about normal flora being on your skin, in your gut, those sorts of things. How does it provide you with that microbiological barrier? What's it actually doing, having normal bacteria present?
Fighting off the bad bacteria.
Fighting off, but how? How does it stop it? Not sure. Competes for space and for nutrition.
Oh, I have
that
that written and then I thought it didn't make sense.
That's what it does, right? So it lives on you, and it's that symbiotic relationship we talked about yesterday. It has a protected environment. It has access to the nutrition and shelter that it requires. It helps protect you against invading bacteria, because an invading bacteria needs to attach and it needs to feed. If there's already stuff living there and feeding, then it makes it harder for the pathogen to get a foothold. Does that make sense? Yep. So that's the answer to that one. And we talked about this one. We didn't go all the way through it. We did talk about B part 1 and what that would mean and what we were talking about. So salmonella contain enzymes crucial for metabolic function. One of the enzymes is ATPase, which breaks down ATP to form ADP and releasing the phosphate. So hopefully we're really familiar with that kind of reaction anyway as a result of cellular respiration. Sorry, not of cellular respiration, but of ATP being used after cellular respiration and that cycle. We touched on this already, so hopefully you answered this yesterday, so I'm hopeful that we're still okay with it. What's the chemical barrier that salmonella occur in the stomach? Or yeah, a low pH because of just the stomach acid. Yep. And explain the effect of the chemical barrier on the structure and function of ATPase and the ultimate consequence this has for the salmonella. Right, so this is worth 3 marks. So again, I know we talked about this yesterday, but what's the question really asking you about? Is it asking you about salmonella?
No.
No. So what is it asking you about?
The effect of low pH on the structure and function of the ATPase.
Yeah, or just of enzymes in general. Yeah, the enzyme itself, they've given you a specific example. Doesn't really matter. Like the example doesn't matter. It's just asking you about the effect of pH on enzymes, which you know, right? You know what the effect of that is. So the first thing is that you would say that the chemical barrier has a low pH, which will do what to the enzyme? What does it actually do to the structure and therefore the function of the enzyme?
It makes it. Does it denature it?
It does, absolutely. It denatures the protein. Yep, because it'll be outside of its optimal range. So it will denature the enzyme. What does that do to its function?
Doesn't work.
Yeah.
As quickly.
Basically breaks down its function. So that's the second mark. First mark is saying that what effect does it have? It'll have a pH that's outside the optimal range. A low pH is likely to cause denaturation of the enzyme, changing the shape of the active site, meaning that it can't function. What is, and that's 2 marks. What is the ultimate consequence for the salmonella? What does that mean?
It'll die.
Yeah, it'll die. It can't break down ATP, it'll die. Yep. So most salmonella actually, if you ingest salmonella, most of the time your stomach acid will actually kill it. Most of the time. It obviously doesn't always work, which is why we still get salmonella food poisoning. But again, this is just relating a topic that you're now learning, and you'll get questions like this in the exam where it asks you to unpack, yes, the initial pathogen stuff or stuff that you're learning about chemical barriers or microbiological barriers, which are both in this, and that's what we're learning at the moment. But it's going to, within one question, part A, B, C, it'll throw back potentially to enzymes. Or fast forward, which we haven't done yet, and talk about antibacterial agents versus antiviral agents and stuff like that. So there'll be stuff like that coming.
What do I put after it denatures the enzyme changing the shape of the active site?
Which reduces the function of the enzyme. Right, because if the active site is changed, then it can't bind to the substrate. And ultimately, if it can't break down ATP to release its energy, it'll have no energy to drive its metabolic reactions, it'll die.
Thank you.
All right, so question 19. So in response to the growing need to provide specific and targeted therapies against specific pathogens to ensure their effectiveness, many new technologies are being developed. For example, radioactive primers which can fluoresce under UV light provide an effective method of identifying viral infections. Adenoviruses, which are a group of DNA viruses, can be detected using radioactive primers. To be honest with you, all of that is just background information and not particularly relevant to what your the answers to the questions. The fact that it's adenovirus doesn't really matter. The general structure of a virus, how would you describe that? So we looked at that yesterday. Let's go through the various steps. There's two categories again, cellular and non-cellular. So let's start there. Is a virus cellular or non-cellular?
Non-cellular.
It is non-cellular. All right. So what's the general structure?
Non-cellular.
Yeah, non-cellular, but what is it? So the two non-cellular, you've got prions and you've got viruses. Only one of them has genetic material. So is that a virus or a prion that has genetic material?
Virus.
Viruses do. And it actually kind of gives that away here. Adenoviruses are a group of DNA viruses. So it means that the genetic material for your adenovirus is DNA. Is it just naked DNA floating around?
No.
Nope. So what is it?
It's coated in, I can't remember what it's called.
Yeah, but just in general, what's it made out of? Even if you can't remember the name is capsid, but if you can't remember the name, what's it made out of? And you know, it starts with P. We talk about it a lot. Just protein. Don't overthink it. It's a protein coat, right? So it's a protein coat wrapped around the genetic material. And that coating is called a capsid, all right, a viral capsid. Explain whether or not viruses can be cultured on nutrient agar. All right, so you're using an agar to grow it. Will a virus grow on an agar plate?
No.
Okay, correct. But the question said explain, so why not?
Because it needs to be like attached to something so it can insert its DNA into another cell.
So insert its DNA into what?
Into other cells in the body.
Into a cell. Yeah, it's remember viruses are intracellular parasites. There's no cells in nutrient agar. It's just agar. It's like a jelly. So it's not going to grow on nutrient agar because there's no cells. A virus on its own can't do anything. It's a parasite. It needs to be inside a cell. Cool. Hopefully this bit will be okay with. Identify one physical barrier that prevents the invasion of a virus. So a physical barrier.
Skin.
Yep, absolutely. It doesn't say what virus. So, you know, typically COVID, for example, is not going to enter through your skin anyway. That's not how it gets in. HIV, though, wants to get into your bloodstream. It will enter through the skin. If your skin is broken, you can potentially get HIV through the skin or through the break in the skin. So it does absolutely protect you. Remember, we talked about that yesterday, skin being one of the most effective barriers you can possibly have. So, yep, skin. The other one would be things like mucous membranes. Like the membranes in your nose and your eyes and stuff also act as a physical barrier. This last one is a throwback to stuff that we've talked about before. A radioactive primer. What do we mean by a primer? So I want you to piece together the pieces of the puzzle. Adenoviruses are a group of DNA viruses. So we know that it's going to talk about genetics, it's DNA. That's sort of your hint when we start talking about primers. Where have we talked about primers before? Does the phrase sound familiar at the very least?
Yes.
That's a good start. All right.
It's something
So-
to do with DNA, but I don't know if it's like
It's-
manipulating it or duplicating it or what.
Yeah, or amplifying it. What's the name of the process used to amplify DNA?
Translation.
No, it's an artificial process
Ah,
that we
Is that-
use.
safe.
PCR.
PCR, yeah.
Ah,
So the polymerase chain reaction. So remember with the polymerase chain reaction, can you remember the steps of PCR? So the first step is denaturation, which does what? We're going to do some revision before the exam. Okay, so remember denaturation splits the two strands of DNA,
mhm.
right? Because DNA is double-stranded. The adenovirus will have two strands of DNA. You split it. Then you do annealing. What is annealing?
Putting the primers on.
Yeah, you attach the primers. If those primers are radioactive, it means that you can then use sensors to detect the radioactive primer that's attached. So you've got annealing and then you've got extension. All right, so you attach the primer and then it builds the complementary strand of DNA. You run that over multiple cycles. You end up with multiple strands of the DNA. In between, there's wash steps. If there's nowhere for the primer to bind, right? So if there's nowhere for the primer to actually bind to the DNA, when you wash it, you wash away all the radioactive primer. So when you run it through the detector, there's nothing to detect. If adenovirus DNA is present, the primer will bind, it will amplify up. When you run it through the detector, it will detect the fact that the primer is still present. Does that sort of make sense? So that's really what it's asking you. Yes, they're tricky questions, right? But they're not unrealistic for what you might encounter down the track. They are open book when I give you these to do. So you can absolutely look through stuff. But the question is, like this here, I would suggest at the moment we're at, and it's okay that we're there at the moment. I'm not stressed about that. I will be if it's a couple of months down the track. But your goal still is, like if I ask you about D, were either one of you confident that you could relate how the primers then go back to PCR and that could then relate to how you could detect stuff?
No.
As we've talked through it, does it all sort of click in your mind and it makes sense?
Yeah.
Yeah. You're being very silent there, Paige. Does it make sense? Not really?
Not really.
Okay. We'll keep working on that. All right. But where we need you to be able to get to is that you can read that and go, primers, I know what that is. That's related to binding to DNA. Oh, yep, that's PCR. And then now I can answer the question. Because when you get prompted,
Yeah.
Yeah. when you get prompted and when you get directed a little bit with the questions, you guys are then able to unpack it a little bit more.
Sorry.
But at the moment, where you get stuck is on that initial phase, and that's not going to help you. Like the reality is that out of these, 1, 2, 3, 4, 5 marks, how confident do you feel you get any of those?
None.
You reckon none?
Mm-hmm.
You would have known the virus wouldn't grow on nutrient agar.
No.
No, you didn't feel confident with that. Okay. Physical barrier, I reckon you could have got. Name some physical barriers for me. Oh, Paige, name one for me. Even if it's the one we just said, I don't care.
I can't remember what you just said.
One physical barrier. So what's a physical barrier stopping infection getting in?
Hairs in your nose.
Yep, fair enough. That'll do. That will get you 1 mark, right? Now, that is, in fairness, we only learned that yesterday. I don't know how much revision you've done between yesterday and today. Plus, you've had other classes and stuff that all cloud your head. So I'm not going to get stressed about that at this stage. That's new knowledge that we're still working on. But don't let it overwhelm you. Like it's going to be, we're going to look at ways that we can consolidate that. And some of the holiday homework that I'm giving you is largely about that consolidation. All right. These questions, though, are good because there's 2 reasons they're good. One, when you look at the back of your textbook, they walk you through the answers and they tell you there's literally a checklist to say, yes, I've included that. Yes, I've said that. Yes, I've done that. which is going to help you go, okay, well, yeah, I understand that this question relates to PCR, or this question relates to enzyme activity and pH, or your first step is to understand what the question wants from you. And that we've identified that's an area that is probably causing the biggest barrier to you guys doing as well as what you can. So we're going to have to keep practicing that skill. It'll get easier. The fact that it's hard at the moment is okay. We just don't want it to be hard sort of mid-October. We want it to start being a sort of second nature thing for you guys. So we'll keep working on that. Is that all right? Cool. All right, you guys happy to have a bit of a look at the next chapter? Haven't read the chapter, I assume.
No.